Now I will speak about autoimmunity. So we'll write. So the meaning of auto means self, immunity. So what is this? Normal immunity and autoimmunity. See, normal immunity is nothing but the immune cells which are now released only during the actual infection. Okay, when any type of the bacteria, virus, which are like deadly microbes, enter into our body, at the time the immune cells will be released. Okay, to just to neutralize them. That is immunity. What is this autoimmunity? See, for example, if this square is the body, like human body, and let us take okay, so this is now bone. Okay, so this is now the bone, the part of the body which normally contains so many cells. Obviously we know that. Okay, each and every part of the body is made up of cells itself, but different types of cells. That is based on their function. Okay, now this, let us take that we have got immune cells. We have got like very important immune cells, B and T lymphocytes. Okay, so we just write this as B lymphocyte and this is T lymphocyte. In our body we have specialized lymphocyte, that is T lymphocyte, which normally plays an important role in determining the self cells and non-self cells. It can differentiate between self and non-self cells. Okay, so we just write here self cells. What is the meaning of self cells? Self cells means the cells which are present in this square, that is the body. Got it? Whatever the cells I have, that is my self cells. See, the primary function of these T lymphocytes, T is to kill these cells. Okay, remember it. It is going to kill these cells. What kind of cell? Non-self cells. So, what is the meaning of non-self cells? It is nothing but the pathogens. Obviously, it is a living organism, right? It must have cells. Okay, when this pathogen enters into this square or body, obviously the T lymphocyte will just go and kills it. Okay, it's like there are so many processes. Okay, what if this T cell is now mutated? Okay, you know about the mutation now. Something has happened in the DNA of the T cell. Okay, it is mutated. Now, instead of differentiating the self cells from non-self cells, what if it fails to differentiate now, which leads to the killing of our own cells? It happens. In like, let me tell you, 30% of the Indian population, they have this disease, okay, which is called as arthritis. Yes, you have heard about this arthritis. It is nothing but kind of inflammation of the joint. Okay, why? Is it because of a pathogen? No. Is it because of some chemicals? No. Is it because of the mutation in the bone cells? No, nothing else. It's mainly because of degeneration of the bone cells. Degeneration means the death of the bone cells. Okay, which makes the bone weaker. Why the degeneration is happening? The degeneration is happening because of mutated T cells. When the T cells fails to understand which is exactly self cells or non-self cells, done. So it attacks both non-self cells if it is present. At the same time, it's going to attack our own cells. Got it? For this, what is required? How we can prevent autoimmunity? Okay, I forgot to tell you. So this mechanism, if a person now suffer from a disease because of improper functioning of the T lymphocytes, that is, it's going to kill our own cells, then it is called as autoimmune disorder. Please note this point. Autoimmune disorder. Autoimmune disorders. Good example for autoimmune disorder is rheumatoid arthritis, which is nothing but degeneration of the bone cells. Even osteoarthritis is also a very good example for autoimmune disorder. Got it? Why? Because the T lymphocytes is going to degenerate our own cells. This is the thing, it's very dangerous. Okay, so what is the reason behind it? The mutation of T cells, lymphocytes. B cells are now helpless, they cannot do anything else. Okay, now let me just tell you about the different types of T cells. Okay, I just let me add the different types of T cells. Mainly T lymphocytes, stay normally originated from the bone marrow and they migrate to the thymus, hence they are called as T, thymus. Okay, then they get differentiated there, then they enter into our bloodstream and they will be like, flowing here and there within our bloodstream actually. Okay, see this T cell again differentiate into 4 different types. Okay, in T cell there are 4 different types. What is the first type? The first type is called as TC cells. What is this TC? Very, very important T lymphocyte which now is going to kill a cell. Okay, so the meaning of TC is cytotoxic. The meaning of cyto means cell. Toxic means dangerous to the cell. Cytotoxic T cells. Note this one, TC cells. Okay, very, very important during immune response. The second one is TH cells. What is the meaning of H here? It's going to help the TC cells, the main function of TH cell. Okay, normally TC cells are like direct in action. Okay, whenever they see the pathogen, if it is presented by the antigen presenting cells, APC. Name some APCs. It could be B cell, it could be dendritic cell, or it could be, I think, the macrophages. BMD, BDM, whatever it is. Okay, so they are going to present the antigens to the T cytotoxic T cells or TC cells. Okay, these TC cells, they have TCR. What is TCR? T cell receptors. They are going to bind to this and then they neutralize this. This is actually the mechanism actually. And for this mechanism, we need these help. So cytotoxic cells require the help from another type of T cell, that is TH cell, that is called as helper T cells. H means helper T cells. Normally like the HIV virus. First thing is the HIV virus is going to bind to helper T cells. See, remember it, TC cells, they cannot work on these antigens if it is presented by APC independently. They need the help of TH cells. That means for the activation of TC cells, TH cells are very, very important. If we just inactivate these TH cells, what's going to happen? These TC cells will not be activated. Got it? So this is actually the principle. What is AIDS? Acquired immunodeficiency syndrome caused by a virus called as HIV. Yes, this HIV will go and bind to TH cells, making it inactive. Done. Okay, and as a result, it cannot help the TC cells. What will happen? The immunity decreases. The person is now prone to get any type of disease. It could be cancer also. This is the thing. Okay. Next is, okay, now let us talk about this TC cells. See, as I said, these T, particularly in T cells, TC cells can differentiate between self, that is our own cells, and non-self, that is foreign cells, like pathogen. Okay, let me just write P. This is our own cells. They can differentiate it. Okay. The main function is they just go and eat up, like kills the cell. Okay. So this TC cell normally, like the main function is it's going to kill the non-self cells, new cells. Okay. But some cases, because of the mutation, that is autoimmune disorder, the TC cell can also kill self cells. This is the thing. So we need somebody to keep an eye on the activity of the T cells, TC cells. Okay, somebody should be there to monitor TC cells. That is called as suppressor T cells. Suppressor T cells. These are specialized type of T cell which is going to monitor TC cells so that they will not kill self cells. This is the thing. Suppressor means, what is the meaning of suppressor? Somebody suppresses TC cells. Okay, so somebody who is actually will take care of the function of TC cells, that is suppressor T cells. Okay, now let us imagine one condition here. So here we have got a pathogen, and now let me just tell you one more point. I thought of making a different video session, but I will continue with this. Okay, so this is now pathogen, a non-self cells, antigen. Okay, so instead of pathogen, let me just write it as A. A means antigen here, which is now in the body of a particular human being. And where exactly the antibody, see, antibody must bind to this antigen. This is very, very important. Okay, where exactly the antibody will bind to this antigen? See, look at the structure of the antigen. It could be like this, or it could be like this irregular, or it could be like a square, or it could be like a linear structure. The shape and size varies from like a type of pathogen. Okay, so let us take this as a good example. Circle. The shape of the antigen is now circle, and here on their surface they have got a special chemicals. Normally they are receptors. That means receptor in the sense they are capable of binding with some other what we say receptors. Okay, so they can bind. And these structures which is present on the surface of the antigens are called as epitopes. Please remember it, the name. What are epitopes? Not for your board examination. Epitopes are nothing but these are special receptors or chemical present on the surface of the antigen. They play an important role in antigen determinants. Please note this point. Antigen determinants. Hope is visible. What is this antigen determinant? So, epitope is nothing but antigen determinants. Please note this point. Epitope are nothing but the chemicals which are now present on the surface of the antigen. They are also called as antigen determinants. Okay, so simple way. Now, we'll erase this now. Okay, in a simple way, I can write this as. Okay, so square is now our antigen, and this is now the epitopes or antigenic determinants. This particular antigen is now within our body. We need our immune cells to detect it. But it's like, who will detect it? This is very interesting. There are so many cells, they are involved in detection first. They detect it, then they must bind to the antigen and take this antigen to the cell. Okay, which will take care of destroying it. Okay, so the first thing is we need something like a component of our immune cell or immunity should bind to this epitope. Okay, or bind to this antigen. Now let us take one good example, that is, okay, so this is now B cell. B cell is also a type of antigen presenting cell. So why I have taken B cell? Because B cell contains antibody, so it will be easy for us to understand. And this B cell contains Y-shaped structure on their surface, that is nothing but the antibody. Yes, okay, that's one antibody. So this antibody, now the B cell now goes to the antigen, and this particular, see, even though B cell contains thousands of antibodies on their surface, but only one will be specific to a specific epitope. Okay, so let us take this is A antibody, this is D antibody, and this is E antibody. So this A antibody is going to recognize a specific epitope. The same A antibody cannot recognize another epitope present on the same antigen. Got it? So they are very, very specific. Okay, so now this particular antibody will now bind to this epitope. Okay, epitope present on the surface of the antigen. Done. So this is binding. This is binding now, and this B cell now, after binding, it will take the antigen to TC cells, or it could be the neutrophils, or maybe the phagocytes, okay, or macrophages. Like that, it varies actually. Okay, so they will take. This is very, very important. So here, what I am actually trying to tell is epitope. Epitope is nothing but antigenic determinants. That means the particular chemical present on the antigen will determine what kind of antigen it is. Got it? Okay. Or like what kind of reaction it is going to produce with a specific antibody. This is the thing. Note this point. Okay. So very, very important. Note this point. Even though the same B cell may be containing different types of antibodies, a specific antibody will bind to a specific epitope. Antigenic determinant. Got it? So this cannot bind to this. This is very, very important. And now the B cell now acts as a antigen presenting cell. So this will now bind to this, and now actually they enter into bloodstream, and finally they present this to this cell. That's what I said, like T cells normally are dependent, not independent. They are dependent. They need the help of somebody like APC, antigen presenting cell. Good example is B cell. Okay. Even the same thing is applicable with the macrophages. Okay. So this is now the macrophages. It has got some receptors on its body. Okay, there are so many things actually in higher classes you are going to study, like MHC1, MHC2, CD8, CD4, so many things. Okay, these are all these specific protein structures. They act like a receptor. That means this is no macrophage. Don't worry. Okay, not required. And this is now the antigen, and it has got epitopes on its body. Okay, so here a specific receptor present on this macrophage cell is going to determine a specific epitope. So then they bind to it and present it to TC cells. Even it is applicable to the other cell, that is dendritic cells. Same mechanism. Okay, all these are now antigen presenting cells to the T C cells, cytotoxic C cells. Okay, remember it. Okay, so now last one, the fourth type of T cell is memory cells. Okay, what is this memory cells? Okay, so we take one example here in this thing. Okay, so this is now, okay, is it visible to you? Shall I erase this portion? Okay, I know that you are not here. So this is now B cell. This is now a specific antibody, and this is now antigen, and this particular structure is epitope. Okay, which is already now bound. Okay, B cell will now present this to a type of T cell. It is not like all the time T cells. It could be neutrophil also. Okay, it depends. But I am taking T cell, and here what's going to happen is, when the T cell, let us take, this is the first time when the T cell is meeting a special antigen in its lifetime. For the first time it is meeting. Okay, it's going to meet this. So this T cell, we just write here. Now T cell will come very close to this T cell where it is holding the antigen, particularly in the site epitope. So here, this particular, the other site of the antigen also contains epitopes. The epitopes are everywhere, okay, but the chemical composition of the epitope varies from epitope to epitope in the same antigen. And this T cell now binds to this epitope by using its TCR, and at the same time something is going to happen. That means actually another receptor will bind to the antigen on its another surface. It's a complex mechanism, and it's going to now undergo. So as a result, okay, so when it now binds, in this case, it's going to activate a system which is called as autocrine. So what is this autocrine system? Autocrine means activating the same cells by itself. This T cell is alone now. It needs companions. Okay. So this T cell now releases some chemicals, that is cytokines and interleukins and so many things. Okay. These chemicals now brings the cell division of the T cell. See, when exactly T cells divide? After differentiation, see, there is nothing. If I talk about during the formation of T cells, if they undergo division, there is nothing actually. Obviously, all the cells will do the same thing. But after differentiation, see, it is a mature T cell. After meeting the antigen for the first time, it will undergo division. Okay. So, in that, one cell will now complete this action. That is killing, removing or neutralizing this antigen. Okay. The other cell is called as memory T cells. See, this is now memory T cells. What is the function of this memory T cells? They are produced because of T cell and B cell interaction. That is interaction between antigen and T cell. Okay. So anyhow, like this T cell will fight with this. At the same time, it's going to die. At the same time, the antigen also will be neutralized. Done. But this T cell will be produced, which will retain the memory of this event. So that is T memory cells, the fourth type of T cell. And now this is not secondary immune response. That's it. Got it? So this is the thing. And when the same antigen enters into our body for the second time after some years, these T memory cells will be activated. Okay, so the mechanism will continue like that. So this is the thing. And they are ready now. These memory T cells are ready to kill this particular antigen if they enter for the second time. So that's all about this. Autoimmunity is nothing but it is the condition where T lymphocytes able to differentiate between self and non-self cells. If they cannot differentiate between self and non-self cells, then we will suffer from a disease called as autoimmune disorder. Hope you understood it.